Hey students, I should have recorded some lectures for you in modules 1 and 2. So we're going to pick up with module 3 and we'll do these from here on out. But we're going to talk about how do we get energy from food. So we'll talk about some cellular processes. There's some terms in here. I hope this video helps you understand a little bit better. But we're talking about cells. And in the world, there are two types of cells, prokaryotic cells and eukaryotic cells. And it's only those two options. Prokaryotes lack a nucleus and organelles, and really the only prokaryotes that exist on the planet are bacteria. Everything else is eukaryotic. So plants, fungi, protists, and animals are all eukaryotic. And they have stuff inside of them, like membrane-bound nucleus, we can see here, organelles like mitochondria, endoplasmic reticulum, Golgi bodies, all of these things. And we're talking about how we get energy from food, and this conversion of energy from the bonds in food into a usable energy that we call ATP happens in two places: the cytoplasm, which is the fluid portion, and then the mitochondria itself, which are these little organelles that exist inside of the cell. Some of the organelles are listed here that we can find inside of a cell, like the nucleus, the ribosomes. This is a nice little table to look at when you're studying what the organelles are and what they do. And this is a great image here so that you can see what they look like. But let's look at the mitochondria itself. Mitochondria uses oxygen from the air. It's the reason that you inhale. It's the organelle inside of your cell that requires oxygen. It's the only organelle that requires oxygen in order for it to perform its function. And what it does is it takes bonds that are holding our food together, and it breaks those bonds apart. Now, when you break a bond apart, it releases energy. And the mitochondria uses that release of energy from the food in order to generate ATP. And by doing so, it creates carbon dioxide, which is why we have to exhale. The mitochondria itself has two membranes, an outer one and an inner one. Inside the inner one, you have this inner membrane that's kind of folded back and forth to increase surface area. These are called crista. And there are these spaces that exist in here that we call a matrix. The mitochondria is kind of a unique organelle in that it has two membranes. It also has its own set of DNA that is circular, like a prokaryote. Now, there's this endosymbiotic hypothesis for how we acquired mitochondria as a eukaryotic organism, and it's basically the endosymbiosis theory postulates that a large eukaryotic organism at some time in the distant past, when there were only single-celled organisms on the planet, consumed some aerobic bacteria that the large cell didn't digest right away. And the aerobic bacteria were then had a symbiotic relationship. The anaerobic bacteria would generate food for the aerobic bacteria, and therefore, you had this space for the aerobic bacteria to live in, and it increased the likelihood that this anaerobic bacteria could exist in this new world with oxygen in it. And so therefore, the mitochondria just kind of stuck around. So you should be able to answer this question after the things we just talked about. Just kind of check it out and see if you understand which of these items eukaryotes have and prokaryotes don't have. The plasma membrane is this membrane that surrounds the cell. It contains 2 layers of phospholipids. These little things here are the phospholipids. They have a hydrophilic head, which means that they like water, and a hydrophobic tail, which means that they don't like water. And because of this bipolar nature of these phospholipids, this allows them to align the tail so they point inward, and it keeps the water out so that you have this separation of environments from inside the cell to outside the cell. It's like a little wall. The plasma membrane has cholesterol inside it. It helps to regulate the fluid nature of the membrane. It keeps it stable at high temperatures and low temperatures. It's like antifreeze almost. There are proteins in there. These proteins act as little channels so that things can get across the membrane. There are glycoproteins that serve to identify the function of the cell to your immune system, and cytoskeleton proteins that kind of help provide a shape to the cell. And the plasma membrane is considered a fluid mosaic model, as it's fluid because the phospholipids move around, and it's mosaic because there's a lot of components in there. Materials can move through the plasma membrane in one of 3 ways: passively, and it's called passively because it's not requiring energy to do so. Diffusion, facilitated diffusion, osmosis. All of these things move from an area of high concentration to low concentration, and that's what this concentration gradient means. So here we would see there's a high concentration of dye in this area, and this dye would diffuse throughout the whole fish tank until it's spread out and reached equilibrium, so that there is an equal amount of dye and water spread throughout. Now, it doesn't mean that the dye molecules stop moving. It just means that the net movement is 0. Active transport is energy is required to move materials against the concentration gradient. So if you tried to get all of these molecules of dye here back into this one little space, it would take energy to do so. And then you have bulk transport, and that's moving large things like pieces of food to a bacteria, or, and in this case, your cells would wrap them in a membrane and then move them across the membrane. Let's see. Facilitated diffusion is a type of diffusion where substances will move from an area of high concentration to low concentration, but it does so through some protein. Glucose and other molecules are a little bit too large to pass through on their own and are transported this way. Also, molecules that have a charge are moved this way too. Excuse me. Anything that has a positive or negative charge to it, like potassium, sodium, and calcium, require a protein to move through, and so does water, actually. Water is slightly, has a polar nature to it, and so it cannot pass through the membrane itself. Oxygen, carbon dioxide can move directly through the plasma membrane and does not require a protein, and so that would just be called diffusion. And facilitated diffusion means that it diffuses with the help of a protein. Osmosis is the diffusion of water towards areas of high solute concentration. And in the case of osmosis, it's the diffusion of water, but it's the diffusion of water across some membrane, right? And when we talk about osmosis, we hear these terms isotonic, hypotonic, and hypertonic. And I want to point out something to you here. In the case of water moving from an area of high solute, if you looked at this, so in a hypotonic solution, which means that inside the cell, maybe there is like 7% stuff, salt, calcium, whatever, but inside here you have a 7% solute concentration. Well, if it's a hypotonic solution out here in this fluid environment, let's say that it's inside of a beaker or something, maybe out here it's only 2% stuff, maybe a little bit of salt, but there's not as much stuff here as there is here. And so therefore water is going to move from low solute concentration to high solute concentration. And water does that to try to even out the solute concentrations there. And in a hypotonic solution, the cell would actually burst. And here we have the opposite. So here we have a hypertonic solution. Hyper means more solute. So here you would have the opposite. So out here maybe you have 7% stuff. In here maybe you have 2% stuff. And in this case, water would move out. Real world example, hypotonic solution would be like a freshwater fish, like catfish you have here in the Ohio River. They constantly have to produce urine because water is flowing into their cells, and they have to get rid of that water. The opposite is true of marine fish, like tuna. Tuna actually have to actively drink water because they lose a lot of water because they exist in a hypertonic solution. Active transport requires an energy source in order to move molecules against their concentration gradient. Sodium potassium pumping is an example. So if we look here at this, we would see that you have sodium out here on the outside. You have some sodium inside, but there's more outside. And so what's happening here is we want to move these sodium out here, but in order to do so, we have to spend energy to get them out. And when we spend energy to get them out, it's like a pump. It's like moving something like moving something uphill, basically. Well, look at these things. Let me know which ones are passive processes. And we just talked about them. Passive means it does not require energy to work. Well, that brings us to cellular respiration. Respiration uses 4 stages to break down the chemical bonds of organic nutrients like glucose into ATP. This happens in glycolysis is one of the stages. The citric acid cycle is one of the stages. There's a preparatory reaction too right before citric acid, and then you have the electron transport chain. And we only get ATP out of glycolysis, citric acid cycle, and the electron transport chain. This is glycolysis, and it looks complicated, but let's break it down a little bit. It's the first step in cellular respiration. It occurs in the cytoplasm, just the fluid portion. Glycolysis has 2 main phases. All right, the energy investment phase, which means that we spend energy. And now, here we have glucose, and glucose has 1, 2, 3, 4, 5, 6 carbons. And we need the energy between these carbons, which are held in these little bonds. So the energy for this glucose is here, 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 and here. And we need those bonds to break. And if you break them, you can release the energy from the glucose. Well, we have to spend 2 ATP in order to break it into 2 pieces. And these 2 pieces are called G3P. Well, then we get into the energy harvesting steps, where you add another phosphate, and now we make this thing. This NAD plus is an electron carrier, right? It's like a little dump truck. And just like real dump trucks will be empty. They'll get filled with something. Those dump trucks will then drive somewhere else, offload the material, and then they'll drive back to the work site. And that's exactly what NAD plus does. NAD plus is an electron carrier. When it's NAD plus, it's empty. When it picks up electrons, it becomes NADH. And now we have a full dump truck. This dump truck will then drive, or it will go somewhere else in order to drop off the electrons. And I want you to remember that, because we're going to see where this NADH drops its electrons off. Here we see the BPG kind of continuing on. It generates an ATP here, and it generates another one there. And the same thing is happening on this side as well. So it happens kind of simultaneously. So we spent 2, we made 4, so we have a net gain of 2. So we generated some ATP, but most importantly, we also generated some electron carriers. Pyruvate, which is what the ending molecule is here, will then go through the prep reactions. So here we see glycolysis turning glucose into pyruvate. We generated some ATP and some NADPH. And the prep reactions occur in the mitochondria. This pyruvate will then go through the citric acid cycle. And this citric acid cycle requires this pyruvate acetyl-CoA to go into it. And when it goes into it, it will tear some more bonds apart. And by doing so, will generate some more ATP. But if you look, what else this citric acid and prep reactions generate is some more NADHs, which are electron carriers. And then you have this thing, which is FADH. It's an electron carrier too. It's built a little different, but it's kind of like the difference in a yellow dump truck and a green dump truck. They both do the same thing. They just kind of look a little different. Well, again in the mitochondria, we have this thing called the electron transport chain. And this is when we generate most of our ATP. Remember, we've created a lot of NADHs. These NADHs will then bring those electrons. And so here we see the NADH there. We will bring those electrons to the electron transport chain. And the electron transport chain consists of these 3 kind of major protein complexes. And these electrons will move across. And as they move across, those electrons power on these protein complexes. So they basically turn them on. And these protein complexes are just proton pumps. If you look here, you see a proton. This proton will then move from inside the inside membrane or the matrix to this inner membrane space. We see one here and one here. And the reason that they're moving across is because these electrons are moving down. Oxygen sits down here at the end, and it will accept these electrons, and so it's the final electron acceptor, and it generates water. And so we kind of create water with this with the electron transport chain too. Now, because we have pumped all of these protons out here, we've created a concentration gradient where it's high outside and it's low inside. These protons don't want to stay out here, and so they will then flow back through this other protein called ATP synthase. And ATP synthase will take the energy from these protons flowing through like water and generate ATP from it. And if you look, we generate around 32 or 34 depending on this, and we won't get into that. But here we make a lot. We only make 4 ATP with glycolysis and citric acid cycle, which is really small compared to the large amount we can make with that electron transport chain. So a single glucose molecule can make 36 to 38. A single fat molecule can generate up to 108 ATP molecules, but they take longer to process. Protein can also be used as an energy source with amino acids. But if you use amino acids, they have to be deaminated by the liver before being used to generate energy. And that process can cause some issues biologically too. So we don't really want to use our proteins for energy, but we will, our bodies will, if we don't have any other options. So that is the end of this module. I hope this video helped kind of explain this material to you.